So, good morning. It's my pleasure to chair the next session together with Sally. We a long time ago met in Seattle already while you were doing your postdoc there, and I started my academic career also in the field of biomaterials. And I think this was a very delightful start, Dave, of this meeting, because I think it's up to you as the new generation in the field to ask very critical questions. And your provocative start should encourage you now all in every single talk to ask yourself what has been shown in cell culture, what has been shown in mice, and what has been validated. Because there were certain topics over the last 30 years that were all kind of huge promises were made and finally were not realized in the clinics. And initially, most people in the field started out with a chemistry or chemical engineering background. And I think you now, if I look around here, many of you have very diverse, different backgrounds. And so we should also ask the question in every single talk now this week, are there certain things that the community so far has forgotten to look into that are important to solve the questions that Dave demonstrated here? So the next session is called Regenerative Medicine, Stem Cells and Forces. And it's my great pleasure to introduce Molly. And Molly has been a chemist by training. She got her Ph.D. in Polymer Science in 1992 at Amherst. She then, and that makes her different from many bioengineers, you joined industry, but at the same time you had an adjunct professorship at Brown University. And then, so she had this industrial experience, came back to academia, then started her academic career in Toronto in drug delivery, and she focused on the brain and the spinal cord. She got many awards along your way. And I would like just to mention a few: the L'Oreal UNESCO Award for Women in Science in 2015. She was made the Inventor of the Year, an honor from University of Toronto in 2013. And then there are a few other nice awards since then. So, Molly, we are very much looking forward to your talk.
It's all about tag teaming today. So I'll introduce Dave, and then you guys can work your way through maybe how you're going to do this presentation. Is that okay? Yeah? So it's always that challenge when you come together on the day to work out how you're going to do this. I'm Sally MacArthur, for those of you who don't know me. I'm from Swinburne University in Australia and CSIRO, which is Australia's government research organisation. And I'm delighted to introduce Dave Mooney from Harvard University. So Dave holds the Robert P. Pincus Family Professor of Bioengineering at Harvard, but his education base is in chemical engineering, both at MIT and at the University of Wisconsin. What I really liked with thinking about what Dave does is the interest base he has, which is answering into so many of the questions that Dave Granger, or Grave Danger as we've got to know him, has discussed before. So the questions about how do mammalian cells receive information from materials in their environment, which is obviously key. A design and synthesis of new biomaterials by looking at those sort of signalling pathways, and then that regulation and the way the cells then respond in those types of environments. I'm going to stop and let Dave get on with his talk. Thank you, Dave.
So I'm going to give some background information on a topic in terms of looking at the role of mechanics in biology and stem cell biology, and I think this will be complemented by Molly. We'll focus more on the chemistry aspects and some of the signaling molecules and their role in terms of stem cell behavior as we look towards regeneration. Now, this slide was going to be a quick overview of the stem cell niche, and the point that I'll make with this is that Molly's going to talk a lot, I think, about a lot of the chemical signals, the morphogens and their role in stem cell behavior. And so by contrast, instead of talking about a number of these issues, instead I'm going to focus on the role of mechanics. Mechanics are a key issue and aspect of the microenvironment, all the way from stem cells during early development through the stem cells that exist in the adult body. And as a kind of an overview of this issue, I want to make a few points from this slide. First of all, that cells are continuously generating and being subjected to forces generated by themselves, other cells, and more broadly, the external environment. So through actin-myosin interactions, cells actively generate forces. With these forces, they then pull on adjacent cells via cell-cell adhesions such as cadherins. They actively pull on the substrate to which they're attached, for example, using integrin matrix adhesions. The resistance that they feel to these forces to which they apply to the material will be dependent on mechanical properties of that surrounding tissue and the surrounding cells, and the cells will actually sense and respond to the very mechanical properties. Now, there are also obviously a variety of external forces, tensile or compressive, shear stresses that arise from fluid movement across cells, and these are sensed by, for example, the primary cilium. We know that there's mechanically active ion channels that are very important in the response of cells to these types of mechanical cues. If we look just a little bit more closely at one particular type of mechanical signal, that related to when cells are basically pulling or against their adhesions, we know that when cells attach to, for example, RGD ligands present in extracellular matrix molecules, they use specific integrin receptors that form into things called focal adhesions. And there are specific enzymes, for example, focal adhesion kinase, FAK, that when cells basically pull on the substrate to which they're attached, that will impact the size and the formation of these focal adhesions. That will lead to changes in signaling pathway of things like MAP kinase, the Rho signaling. And a key feature here is there's often mechanical feedback. So you pull on a cell, it senses it, and then it does something different. In this case, as Rho gets activated, it can upregulate ROC, which increases contractility, and the cell may, if it feels some resistance, it may then pull harder, for example. That also, these mechanical cues also lead directly to activation of a variety of transcription factors that can then enter the nucleus and directly alter transcription at multiple levels. So cells have the ability to generate and respond to mechanical signals through a variety of mechanisms. Now, I'm not going to focus today on kind of the intracellular aspect. Instead, I'm going to focus on the interface of the cells with the materials and what we know about how the design of that interface regulates the cell response. To put this in context a little bit, I want to go back for a moment to think about development. So we know that mechanical cues regulate stem cells during development. Early on in the embryo, we have very little matrix, so it's mainly cells pulling on each other via cadherins, and these mechanical cues are some of the very earliest signals regulating development. Over time, though, cells deposit a lot of extracellular matrix, as you can see schematically here, and so cells tend to be coupled more indirectly through the matrix. Now, this matrix does a variety of things. It allows for transmission of forces very quickly, if it's purely elastic, much more quickly and over greater distances than one can achieve with purely chemical signaling pathways that require diffusion and thus significant time. The matrix also allows a memory of the mechanical past. As the cells change the matrix that they synthesize and deposit around themselves, that in essence is a history of that cell. And then that information is stored and can impact that cell at later times or other cells that come into contact with that material. Now, some other examples here. We know that, for example, anterior-posterior axis formation is highly dependent on mechanical cues and cell traction forces. Events in morphogenesis are dictated by changes in basically how cells adhere, assemble ECM, and a feature here that, to connect with what Molly will be describing, is that mechanics obviously don't act alone, morphogens don't act alone, the two act together. So for example, in salivary gland morphogenesis, one sees activity of both the mechanics as well as morphogen gradients that regulate formation of structures with particular patterns. Now, that's all kind of very high, big picture and very schematic. To introduce a piece of real data, This is actually a piece of data from a study from 1980, so a very old study, but one that I think is very important in that it started to demonstrate the idea that cells do actively attach and pull on what they're adherent to. Before this point, I think many people in the field had thought of cell adhesion as being a chemical event, but this showed that it was really a mechanical event. And so this is actually some pictures where here is a fibroblast, and then there's a few cells down here. And these pictures aren't out of focus or poor pictures. What they're showing is the cells adherent to a very thin layer of silicon rubber. And the material is soft enough that the cells are actually physically wrinkling the material. So that's what you're seeing are wrinkles in the material. And so this paper was published in Science because it demonstrated that cells can actually pull hard enough to deform the material that surrounds them. Now, after this paper was published, we obviously then began to understand that the mechanism of this pulling related to the cytoskeleton and that the propagation or transfer of those stresses to the surrounding substrate was due to specific types of integrin or other adhesion engagement. Now, once we began to understand that cell adhesion was important, it was a mechanical event, cells pulled on their surroundings, that opened up the question of, do cells then respond to the resistance they feel when they pull on a substrate? So this has been answered using a variety of two-dimensional systems historically. So for example, Yuli Wang was, I think, the first one to really generate convincing data in vitro, where he showed that the migration of cells in two-dimensional substrates was dictated by the stiffness of the substrate. Our group showed a number of years ago that proliferation and apoptosis of cells was regulated by the stiffness of the substrate. And then Dennis Disher showed in what's now a classic paper that the fate of mesenchymal stem cells going down neurogenic, myogenic, or osteogenic pathways was controlled by the stiffness of the material upon which the cells were adherent, and thus presumably the resistance the cells feel to their own traction forces. Now, all this data was in two-dimensional cell culture, but most cells in the body actually live in a 3D world, and so that opened up the question of, do these same types of effects, are they seen in three-dimensional culture? And then, perhaps most relevant to this symposium, can they actually impact tissue regeneration back in vivo? So, since this is a kind of a tutorial type of format today, I want to point out some of the challenges to actually moving the 2D systems, which are pretty straightforward, to 3 dimensions. These include the fact that it's very hard to decouple mechanical properties from a variety of other variables as you go from 2D to 3D. So this includes changes in matrix composition, changes in porosity and architecture that impact things like diffusional transport and degradation of the material. There's also key questions of ensuring efficient transfer of stresses and strains between cells and the material, and then, do the time-dependent mechanical properties also play an important role, which is something I'll get into basically in the last half of my comments today. So if one wants to definitively say that a particular mechanical property is important to cell, you need to actually be able to separate it from other variables in a really clean manner. And the challenge here is shown on this slide, which is the traditional way that people have done these kinds of mechanotransduction studies, is to take purified ECM molecules like collagen and simply to make gels with these at differing concentrations. The challenge is, as you increase the concentration of the protein, for example, in the solution, you get increased cross-linking and you'll get increased stiffness, but at the same time you'll get an increased number of places where cells can attach, and you'll get a decrease in the mesh size or the spacing between cross-links that changes things like diffusional transport of macromolecules. So at the end of the day, you change stiffness, but you also change a number of other things. Now, there's been a variety of synthetic systems that have been developed to try to address this coupling. So, for example, one can take polyethylene glycol and other types of synthetic polymers that don't naturally attach to cells, or that cells cannot naturally attach to, and simply change the cross-linking density using kind of classic, polymer chemistry strategies. And this then keeps the density of ligands constant while you change the stiffness, but you again change the mesh size. So again, diffusional transport is altered as you change the mechanical properties. However, there are systems that one can decouple these. So one of these is a polysaccharide called alginate that we'll talk a little bit more about. And this cross-links via a physical mechanism by sharing of divalent cations between certain units in the polymer. And the basic premise is when you have a lightly cross-linked network of alginate, you have the divalent cations here, these red dots, that are providing cross-links, and the entire region of the polymer ends up getting aligned due to these cross-links. And then as you add more and more, for example, calcium ions, you fill in some of the empty locations, but you don't change the architecture of the network. So the architecture stays constant, so you keep the mesh size the same as you change stiffness. In this case, this polymer doesn't mediate cell adhesion, so it's going to be dependent on providing some other means of cross-linking that you can keep constant as one varies the mechanics. To give you a little sense of what these types of materials we've been talking about, hydrogels, look like, this background image is an AFM image of a hydrogel. So you can see the individual polymer chains, about 5 nanometers or sub 5 nanometers in size. And you can see that most of the material actually is not comprised of polymer, but it's comprised of space between the polymer chains that's filled with water. So this looks very similar to many of the soft tissues in our body, where you have a certain solids content with a very highly hydrated matrix. The key point for today is that one can very readily control the mechanical properties of the stiffness of these materials simply by controlling things like the cross-linker concentration. So in terms of this decoupling that I just mentioned, if one looks at the effective fusion coefficient of a macromolecule, as one varies the elastic modulus in these types of physically cross-linked systems, one can see that a diffusion coefficient stays constant over a wide range of moduli, indicating that we do indeed have a decoupling between mechanics and transport in these kinds of systems. Now, one needs to have a means for cells to grab a hold and pull on these materials. One very common approach to do this is to covalently couple small cell adhesion peptides, such as those containing the RGD sequence that binds to certain integrin receptors, as a side chain off the polymer. And so that's very commonly done to decouple the density of adhesion ligands and the type of adhesion from the mechanics and other aspects of the polymer network. And this allows one to take polymer systems that are otherwise non-adhesive to cells and then become highly adhesive. And here you can see cells attaching and spreading in two-dimensional culture on these kinds of materials. Now, when one takes this approach, one finds that in 3D, similar to in 2 dimensions, that the mechanical properties of the material do indeed regulate the ability of stem cells to differentiate down different pathways. So here you're looking at some mesenchymal stem cells that are placed within hydrogels that are either fairly soft and the cells become fat cells, intermediate stiffness and the cells become bone-like cells, or really stiff, they don't differentiate at all. And this is just a lot of molecular characterization demonstrating basically different facets of this differentiation of the cells. So in 3D culture, as in 2D culture, the mechanics do matter and do control the ability of cells to differentiate down different pathways. This is not just seen in vitro, but also one can take hydrogels with defined mechanical properties, transplant small numbers of stem cells, and see that the mechanical properties that in vitro lead to, for example, osteogenesis, lead to more osteogenesis in bone formation and critical size defects, in this case, in rats. So this concept does apply to controlling stem cell behavior in vivo as well as in vitro. Now, there are a number of facets that one needs to consider as one thinks about these types of systems. There was a really interesting paper published by Jason Burdick a few years ago in which they took these same kinds of stem cells, mesenchymal stem cells, and put them into purely covalently cross-linked hydrogels. And what they found was the differentiation of the cells actually didn't alter as the mechanical properties changed within these gels. And it was only as the gels had the ability to degrade that the cells could sense the stiffness of the mechanical properties. So this tells us that we need to be really quite aware of the interface, and a key question is, what are the actual properties at the interface? If the material is degrading, well, it's going to be degraded by the cells, and that interface may not be very well defined, and so it's actually hard to figure out, is it the mechanics, is it degradation products, is it the change over time that really regulates the process? There's also questions about how does mechanics and topography come into play. So there's a very nice paper that came partially from Viola Vogel's laboratory and then Adam Engler published a paper along the same lines a couple of years later that raised the question of, is it really purely mechanics, or is it partially also the topography, or let's say the material properties at the micro scale that regulate some of these responses? So really, so we really need to think about mechanics also in the context of architecture from that perspective as well. Now, one of the features that's been in common to most of the work in the field until just a couple of years ago, though, was that the assumption was that all we really needed to do was to study this phenomena using purely elastic materials. However, the tissues in our body are typically not purely elastic, but are instead viscoelastic, which indicates that they have time-dependent mechanical properties. So to give you a sense of this, if we do a simple test where you apply a step deformation or strain to a material, a purely elastic material, if you maintain that strain constant, will keep the same stress. You can think of it being like a rubber band. If you take a rubber band and you stretch it, you have to hold it with the same stress to keep it strained. And that's what virtually all the studies had been done in these kinds of purely elastic material. But if you look at fat tissue, liver, brain, collagen gels, you instead see that they dissipate stresses over time instead of maintaining a purely elastic behavior. So that raised the question of, well, if actually the materials in our body tend to be viscoelastic instead of purely elastic, do cells notice, is this actually important? Now, one feature that is important to keep in mind then is the typical covalently cross-linked networks will exhibit a more elastic kind of behavior, while physical interactions, ionic crosslinks, and other types of physically cross-linked gels can actually reversibly bind and unbind in response to application of stress, and so will exhibit more viscoelastic behavior. So one can then create viscoelastic substrates or more purely elastic simply by controlling the type of cross-linking in the system. For those of you that aren't familiar with these types of materials, one way we can very readily characterize whether a material is purely elastic or viscoelastic is by doing a frequency sweep, doing it using a rheological analysis. So a purely elastic material, the modulus of the material will be independent of the frequency of the deformation, but for a viscoelastic material, you'll have a high dependency on the frequency. So this is kind of a simple test one can do to look at to determine whether something is viscoelastic or purely elastic. There's a variety of simple approaches one can take to vary the amount of viscoelasticity a material exhibits in terms of the rate at which, for example, it will relax. So one can, for example, change the molecular weight, create basically spacers, and all what these do is they change the mobility of the polymer chains within the network and will change the rate of relaxation. So here, for example, is just a series of hydrogels that have the same initial modulus that one would measure initially at the point of deformation, but very different times required for the stress relaxation, where here it's quantified as the half time for relaxation, where it goes from about 3,300 seconds to 70 seconds. And if one now takes stem cells and puts them in these kinds of materials, you find that actually viscoelasticity does make a really big impact. So here you're looking at the ability of cells to spread when they're placed inside gels, which they exhibit more rapid relaxation as one goes to the right. And the images over here look just like those you've probably seen many times of when you put cells in hydrogels, which the cells stay completely rounded and don't really do anything. But if the cells have the ability to physically remodel the materials, not chemically, but just physically remodel the materials, the cells can then begin to spread, migrate, and actually will proliferate. And all of this is in a manner that's dependent on the ability of the cells to attach, grab, and pull on the materials, because if there's not the adhesion ligands there, you don't see any of this behavior. The ability of the cells to differentiate is also regulated by combination of the modulus and the rate of relaxation. So here, if you're looking at mesenchymal stem cells differentiating down a fat pathway, becoming adipocytes, you can see that if you culture the cells in gels that are too stiff, none of the cells become fat. If you culture instead at a lower modulus that's permissive for adipogenesis, the amount of adipogenesis is highly dependent on whether the material exhibits stress relaxation and how rapidly it exhibits this relaxation as quantified down here. Now, conversely, if you look at osteogenesis or bone formation as the opposite fate, you see the soft gel, they don't really differentiate down that pathway at all. The stiffer substrate they do, but in this case, only if the gels exhibit rapid relaxation do you get significant amounts of gene expression related to osteogenesis. Now, a question you may have at this point in time, what is the physiologic relevance of this kind of behavior we're looking at? Well, if you look at tissues that come in the body, that come from the body in which regeneration, bone regeneration would be typically occurring, you find they also exhibit this kind of stress relaxation. So here's some mechanical testing of coagulated bone marrow from rats. These are fracture hematomas taken from human patients coming in for revision surgery. And what you can appreciate in the stress relaxation test, they both show a high level of stress relaxation and actually rates that are very similar to what was empirically found to be optimal for osteogenesis in vitro. So those gels are probably rediscovering the conditions that our body usually uses to drive bone regeneration. Now, this also can be exploited back in the body. So here, for example, is a simple study where these cells, again, were taken into gels that had the same initial modulus, but exhibit either slow relaxation or fast relaxation, and the fast relaxing gels led to much greater amount of bone formation than the identical, more slowly relaxing hydrogels. And Sally, could I ask you how much time I have right now? Okay, so I should wrap up in a minute or so. Okay, so, mechanistically, all the same mechanisms seem to be in play in terms of this is all actomycin dependent, well, not all, but it's largely actomycin dependent. We have the role of FAC, Rho kinases in the signaling pathways. However, in this context of stress relaxation, there are a few more mechanisms that also seem to be important. One of these is that if a cell can reach out and grab a hold of material and it plastically deforms, the cells have the ability to remodel the material. And here you can see that by this halo that exists around the cell. This is an image of an individual cell in a rapidly relaxing gel. And this halo exists because the cells remodeled the cell adhesion ligands, and one is getting a fret signal. So cells can cluster adhesion ligands around themselves if they can physically remodel the gels. I'll skip this and just point out that another key feature that seems to be very important, and this is from some work of Ovi Chaudhuri's lab at Stanford, he's shown that the ability of stress-relaxing materials to dissipate stresses is also very important. If a material is purely elastic and a cell is starting to push against it, as a cell, for example, multiplies, lays down matrix, you just build up the stress that the material pushes back on the cell. But if the material exhibits stress relaxation, that energy can be dissipated, and the cells can then go on to, in this case, undergo chondrogenesis or cartilage formation. Couple last points. David Waite's lab has shown, and this is a really complicated slide, but my main point here is just that a lot of this regulation also seems to be related to control over the volume of the individual cells. And that seems to be a very important parameter in all these mechanotransduction pathways I've been describing. And the last point I'll point out is that the vast majority of work in this field has been done on a population average basis, analyzing thousands or millions of cells at a time. Recently, though, technologies have been developed that allow one to encapsulate individual cells in very thin layers of hydrogels, so one can now do all these same kinds of mechanotransduction studies on a cell-by-cell basis and begin to look at the individual cell response in this type of setting. So I'll wrap up there and just make the point that where is this, in terms of where this is all going, we've learned a lot about mechanobiology, and it's increasingly being used in the context of mechanoregeneration, where, for example, a variety of groups have taken various stem cells, putting them on synthetic materials in vitro, and then transplanting those cells to then induce tissue formation in desired states. One can instead simply expose the cells to certain types of mechanical properties outside the body before transplantation, and there's been a variety of really exciting work in that regard. And then, obviously, though I didn't have time to talk about, one can also start to think about imposing external mechanical cues on cells and tissues to promote regeneration. So I'll finish up there, and I'll leave it to Molly now to give the second half of the lecture.
Yes. Okay. Hi. So thanks for your patience, and thanks, Dave, for kicking us off. I think we've heard a lot of really important points so far in terms of validating the models that we're using in vitro, and and looking at the complexity of the material in order that the complexity of the material reflects the complexity of the biology. So one of the things that we're really interested in doing is in our design of materials is to really look to the biology. And I think the reason I wanted, Dave, for you to start is because I knew you'd give a great introduction to the importance of that. And in the first slide that Dave showed, he talked about the intracellular mechanism. And so really used that as the starting point in terms of the design of the material. So what I wanted to do, because our session was called Regenerative Medicine, was take you really into the in vivo situation. And in regenerative medicine, we typically think about stem cell transplantation or endogenous stem cell stimulation, so stimulating the stem cells that are already within us. And our lab is focused primarily on the central nervous system and also in cancer. And I wanted to highlight these questions that I think are the key questions facing the field in regenerative medicine. And they apply both to cell transplantation and cell stimulation. And so if you remember nothing else, if you can remember these two key questions, then I think you will have taken something away from this part of the talk, which is how can we enhance cell survival and cell integration? So most of the cells that are transplanted die. Okay, and so if we are trying to replace lost function with cell transplantation, how do we get more of them to survive? And the other key question is in cell integration. So if we are trying to replace lost tissues and organs, or even just lost function, how do we get those transplanted cells to integrate into the existing host tissue? And we are very much focused on the neural circuitry. But there's this also another key question that has emerged in the field recently, and that is, do we actually need the cells to integrate? Are they simply there providing paracrine effects? So that is a key question that is facing the field right now. Presumably you need them to survive, but for how long? And the reason why question number 2 is so important is because we've seen, I think many people in the field have seen with cell transplantation that they have beneficial tissue and functional effects, but there's still very few cells that they can detect. So how long do we need the cells to survive, and do we need them to integrate? So I'm going to talk to you about these two big questions that we face in the field in the context of some of our own results and data. And I'll introduce you to this material, Hyaluronan methylcellulose, which has some really interesting engineering properties in that it's a gel. It's shear thinning. You apply some force, and as soon as it comes out of the syringe needle, it's a gel again. And so this is just a very small movie just showing that the material is injectable. And so we thought this was really important for cell transplantation to make the surgery as simple as possible and to make the when you're instead of putting a physical implant, putting in just a small needle. So the whole point of this entire video is just to show you that this material, as it comes out of the needle, immediately forms a gel again. So it's very fast gelling. This would be described as a viscoelastic material, as Dave pointed out, because it's just comprised of physical cross-links, and those are through the methylcellulose. We don't actually have pennies anymore in Canada, so I think this always just amuses me. Okay, so then what I want to do is tell you how we've used this material to deliver cells to the back of the eye, to the retina, and this is applicable to some of these major diseases. Where vision is lost due to the loss of 2 cell types at the back of the eye, and these are the retinal pigment epithelial cells. These are these gray cuboidal cells at the very back of your eye, and the photoreceptors, the rods and cones. And this work was done in collaboration with Derek van der Kooij's lab. And what we did is his lab has discovered the retinal stem cells in the ciliary body in the eye, and our lab developed that hydrogel. And in that first study, we used the hydrogel to deliver retinal stem cells right to the back of the eye where they're needed. And this work was all done by Brian Balios, who's now an ophthalmologist, and you can see, he's looking down at the microscope, and a mouse is looking up at him. Not photoshopped. Okay, so the first thing we did when we put the cells in the material was to make sure it wasn't going to do anything bad. And what we noticed was that when we mixed the cells into the hydrogel, got a really lovely distribution of cells in the material, and that's what these data show. The other thing, these show the little green dots are the cells, and there's a very faint cube here, so it's hard to see. I was just at another conference last week, and I was talking with some people at a company, a stem cell biology company, and they're doing cell transplantation. And I said to them, oh, you'd never believe what this other company does when they inject cells into the back of the eye. And they're like, what? And I said, they take the syringe and they shake it in order to keep a suspension of cells so that they don't all aggregate. This is while they're injecting into an eye. And then this other company said, oh yeah, we do exactly the same thing. And I thought, well, that's crazy, because if you just use a viscous material, it will keep that lovely suspension of cells in your syringe. And this also just shows that the material degrades, by the way. And you can see that when we inject the cells so this is into a mouse retina, so you can imagine how small the needle has to be in order to inject cells into the back of the retina of an eye, of a mouse eye. Anyways, when you inject the cells in saline, which is what most people do, the cells all aggregate. Whereas when you inject them in our hydrogel, you get a lovely distribution of cells. And so just from a pure mechanical and simplicity perspective, there seems to be some benefit. But the other thing we noticed is that we saw some biological benefit with this material as well. Now, what we were really interested in doing was injecting the photoreceptor cells and not just the stem cells, because we wanted to replace the actual cells that are dying when you lose vision. And so this just shows you that if you use taurine and retinoic acid, you can get a high population of rod photoreceptor cells from the stem cells. And so these are the cells that we transplanted. The other really important question in the field is, which cells should we be transplanting in the first place? So Dave Granger brought up, this idea of co-cultures and multiple cells, and we have some more recent work where we've looked at that. But the other question in the field is, where along that differentiation pathway should we be transplanting our cells? And so one of the things we did is we looked at transplanting cells at different days along that differentiation pathway, so different levels of maturity, in vitro maturity. And you can see the cells that were fully differentiated at 44 days, they didn't survive at all. And we got the greatest survival of these cells, which are more mature, the 28-day cells, in the hydrogel versus saline, and more than any other pre-differentiated time point. So these are the cells that we went forward with. But most people in the field, and it really depends on the cell type, are working at the undifferentiated cells, and there's certainly good reason for doing that. They're the most plastic, and that can be a good thing or a bad thing. So that first idea, does the hydrogel itself do anything negative to the cells, turned out not to be true. Like the hydrogel really has no impact on the phenotype of the cells. They remain rhodopsin positive. and it has no effect on the stem cells either. So that was good. So what we have then now is we have the cell type that we want to transplant. We have a hydrogel that provides a nice distribution of cells when it's injected, when the cells are injected in the hydrogel in vivo. So we have those two key questions that we need to investigate. And so that first question was all on, can we enhance cell survival? And so I'll show you how the material achieves that. But the other question is, can we promote cell integration? How do we get these cells to integrate into the neural circuitry? So where we're injecting them is right between the RPE and the photoreceptor layer. And what we want them to do is migrate across this outer limiting membrane. So in order to enhance integration, we deliver this small molecule, which is a transient gliotoxin. So what that means is that these end feet, these glial end feet from the Mueller glia, which make up that outer limiting membrane, they retract for a short period of time, allowing our cells to migrate into this outer nuclear layer. Well, that was the hypothesis, and other work by West et al. Had shown that that small molecule would provide that function. So we were really excited when we saw these data. So our cells are in green, so they're GFP labeled, and you can see them migrating into the outer nuclear layer. So that's exciting because this small molecule seems to enhance integration. Now I have to tell you, within the past couple of years, there have been some questions in terms of, in other labs, seeing when you have GFP in the outer nuclear layer, are those actually the cells that are migrating, or is it material transfer? Are we getting transfer of genetic information through the photoreceptors? And so, you know, the great thing about science is we're always questioning our own results. And it's a little bit more complicated with the hydrogel because the hydrogel seems to have other effects than just injecting in saline. So we still think actually that these cells are the cells migrating and integrating into the outer nuclear layer. So interestingly, when we deliver the cells in the hydrogel, we see more cells in the eye and more cells in that neural retinal area when we inject the cells in the hydrogel and the alpha-aminoadipic acid, so that pro-integration factor. So that was exciting, and even more cells than just injecting in the hydrogel alone. The question is why? Why are we getting more cells surviving in the hydrogel? Because all I've told you up to now is that the material doesn't do anything bad. But it seems actually like the material is doing something good. And so the question was why? So we went back in vitro and we looked at these rod-derived photoreceptors, the ones that we had differentiated in taurine and retinoic acid, and cultured them in vitro in the hydrogel or just in saline. And what you can see is that there appears to be a pro-survival effect of growing the cells in the hydrogel versus saline. We didn't see this for the stem cells. We only saw it for the rod photoreceptor cells. So we thought it was HA and can interact with CD44, and so we thought this might be a CD44 HA-mediated interaction. The cells do express CD44. That's not impacted by whether they're cultured in the hydrogel or not, but they do express it, and so that led us to believe that this was in fact that type of interaction. So to test it, we took CD44 knockout mice, and we cultured their retinal stem cells and differentiated them in taurine and retinoic acid. And what we saw was over that same 7-day period, we had now lost that pro-survival effect that we had seen. So we lose that pro-survival effect that we see in our hydrogel. We also lose it in the HA. We never really had it in the MC or the serum-free media, and so this points to that being an HA-CD44-mediated mechanism. So it doesn't change the number of rhodopsin-positive cells, it just changes their survival. And so if we go in vivo and ask, what does that mean? With those CD44 knockout cells, so they look exactly the same, they just don't have CD44. They look exactly the same from a rhodopsin perspective. We lose survival and we lose integration. And so this really points out the importance of survival, which we have with the hydrogel, and integration, which we have with that small molecule. And that really goes to that first key question around promoting cell survival and integration in order to have functional repair. The function actually in this study was done with a very simple study where we're kind of in a dark room right now, right? And so our pupils are larger. And if you go out to the beautiful sunny day, which we'll do at the break, our pupils will get smaller. And so what we did in this study is we shone a very bright light at the pupils of the mice, and we have our internal control. So the right eye has the cells and the left eye has the vehicle, and in the right eye we see the pupil starting to shrink a little. So I'll tell you, this is a really bright light, and if it was a perfectly healthy animal, the pupil would shrink down to about 20% of the fully dilated area, and here we're getting about 85%, so it shows significant improvement to control, but obviously there's further to go. And so one of the things, okay, so now how am I doing on time? Okay, more or less 2 minutes. Okay, so the other story I wanted to tell you related to that second question around do we actually even need the cells to survive in order to see functional repair? So one of the things, we were still on this bandwagon, and we still are, on trying to get more cells to survive, because typically people get maybe 5% cell survival. And so we've been designing materials with growth factors and cell adhesion ligands, some of the same ones that Dave Mooney talked about, to further enhance cell survival. And so I won't go through all the details of this, but I'll tell you very briefly, so I won't tell you how we did it, but I'll tell you what we saw was that when we immobilized this molecule, platelet-derived growth factor, we were doing that on our hydrogel to enhance differentiation of neural stem cells to oligodendrocytes. And we got the same percent differentiation with the mobilized versus soluble factor. The other thing is when we incorporated RGD for cell adhesion and PDGF for cell differentiation on the same hydrogel, we got an enhancement in cell differentiation. And this idea, and I think it was something in one of Dave Mooney's first slides where he showed the integrin receptor and the growth factor closely linked and to cause cell fate determinants. This is something that's also been seen by Jeff Hubbell's lab. So when we took this and we took cells in this hydrogel, these are now neural stem cells, and this is now a spinal cord injury model, and transplanted them right into the spinal cord, we saw some really interesting things. One is that here the hydrogel didn't do anything negative, but it also didn't do anything positive in terms of the increased number of oligodendrocytes. So even though we saw that effect in vitro, we didn't see it in vivo. But there was an effect on the host tissue. So we saw more oligodendrocytes in the rat host tissue with the transplantation of these stem cells in the hydrogel. And we saw more of the host neurons, again, when we transplanted the cells in the hydrogel versus just in media. And we saw a reduced cavity volume. So usually in spinal cord injury, what happens is you develop a fluid-filled cavity, and this is a result of the injury. And so if you have a smaller cavity, it means the tissue has regenerated, and so that's usually a good thing. And so having a smaller cavity is good. And we saw some functional repair, and this is the injury is kind of partway down the spinal cord, so it'd be akin to a paraplegic, where the rats would be dragging their hind limbs. And so we look at hind limb function, and we see some significant improvement. So this is a good example, though, of perhaps the stem cells. Well, we have no indication that the stem cells are actually integrating into the host neural circuitry. And it seems like the beneficial effect is more of a paracrine effect. So just in summary then, we talked about those key questions of cell survival and cell integration, and then whether we even, how long do we need cells to survive or differentiate in vivo to get functional repair? Because I'm going last, I get to acknowledge my lab and funding, although you could have too, and our collaborators. Okay, so I think we're open for questions. Thanks.
Excellent. Thank you both so much for that. That was really good. So questions from the floor? I have loads, but yes.
Thank you so much for your nice talks. And I would like to ask a question about the CD44 mechanism of action that you explained. So do you think that this is specific for your hydrogel, or can be shared with other types of hydrogel as well in your pathological context?
Yes, so there's HA, hyaluronan, is known to interact with cells through CD44-mediated mechanism. So I would think most hydrogels that have hyaluronan as part of the gel, you would see that. You won't necessarily see that pro-survival effect. So I showed you just the in vitro data where we didn't see that pro-survival effect with the stem cells. And we've looked with other stem cells, the neural stem cells, and even though they express CD44, it doesn't necessarily translate to a pro-survival effect. But other people have seen that CD44 HA-mediated interaction, so we're not the first. I don't know if you wanted to comment on that? Okay.
Thank you very much to both of you. I also have a question for Molly on the first example. I think you convinced us all of the survival and integration of the cells into the retina, but the functional outcome, while improving the situation, did not solve the problem. So what are some strategies that you'll be trying now to further push the functional outcome? Okay,
so we are doing a couple things. One is the two cell types that are lost in age-related macular degeneration are the retinal pigment epithelial cells and the photoreceptors. So we've looked at the co-transplantation of those two cell types, and we've seen that survival of one helps the survival of the other. So that's quite interesting, and that also enhanced the functional repair. But still, it highlights that second big question of, do we even need the cells to survive? Because our survival, even there, is very small. So we are also looking at ways to, I guess really what I think now is we want to transplant the stem cells, but we also want to look at the endogenous cells and see if we can make that environment more hospitable to the cells. And so we're also looking at, and I'll talk a little bit about some of our controlled delivery strategies in terms of how do we deliver biomolecules to that site to enhance the survival. So how can we deliver the cells so that they even stay alive through the injection process? Yeah, do you want to? I mean, not necessarily that, but.
Thank you very much. I also have a question for Molly.
Okay, so the next one has to be for David.
Yes.
Okay, good.
So it's about the predifferentiation data. I was quite surprised. We work with bone marrow, mesenchymal stem cells, and for us it's better to passage them as less as possible.
Yeah.
And do you know what happened between day 28 and 44? Because at 44 you didn't find any cells at all, and 28 was the best. So do you have any hypothesis of what could be the reason?
Yeah, so we've just done some work with neural stem progenitor cells, and so this is a paper that we're just submitting. And we saw the same thing, that the progenitor cells survived a lot better in vivo than the more fully differentiated cells. And so these are neurons, and with the more fully differentiated cells, they start to produce processes, and simply the act of dissociating the cells and lifting them off the plates it results in a lot of cell death, actually. And so that actually had really negative impact on cell survival and also on the host tissue in terms of causing host tissue damage. So well, I don't know exactly what happened in the more fully differentiated retinal stem cells. I think it was probably the same thing. I think that those cells, when just that act of lifting them off caused a lot of cell death, and that may be why we had so little survival.
I would have a question.
So maybe in between we should, we received here 2 questions, and there's one: could you discuss how to investigate changes in mechanics with matrix degradation in the presence of cells? And that's, I guess, to you, Dave.
I got a little too much feedback, I think, for that.
If you just like to read it again.
Oh, yeah. Yeah, so the question relates to then with degradation. So with degradable systems, it's very complex, particularly if the cells are mediating the degradation, because then the environment immediately around the cells, the environment immediately around the cells is changing mechanically. And to my knowledge, there's no good way of, in that kind of dynamic setting, to in real time be monitoring the mechanical properties of that interface. So our approach to this has been to use materials that cells can't degrade. So we at least have that as a reduction in the number of variables. But I think an unmet need in this field is to be able to do dynamic mechanical property analysis at that interface between a cell and a material. Until we do that, we can't really do the kinds of studies that the individual here was asking about.
Can I just ask, what techniques do you think would work well? Do you have any sense of what those techniques would need to do?
Yes, so I think, well, just measuring the mechanical properties at the interface is already difficult enough, even if there's no degradation. So, just doing static measurements is hard enough, and there's actually not good techniques for that. So in terms of the dynamic, I don't know. We're not actually even that far in terms of trying to figure that out at this point in time. We're focusing more on the static at this point in time.
There's a challenge.
My question was,
it's related to that. Maybe I ask it like this. Can you comment on tools or techniques that you propose to study mechanics of materials?
So mechanical effects?
What are your favorites?
Oh.
I mean, in the context of viscoelastic materials.
Yeah. So I would say another challenge in this field, maybe I'll answer it that way, is not related to degradation, but the time dependence of the viscoelasticity. So a common issue here, so a cell reaches out, it pulls, the resistance it gets feeds back to the cell, and then the cell will change how hard it pulls depending on the resistance it feels. So we need to be able to measure not just the mechanical properties of the material, but also the traction force the cells are putting on the material. And traction force microscopy is a very well-established technique in 2D and partially in 3D, but there's not a good strategy for doing that in viscoelastic materials. So there's a few groups working on that now, but I think that still is an open-ended question of how do you actually measure these traction forces in 3D and viscoelastic substrates.
Then we have one last question to Molly. as gelling happens very fast, then how is the process of cell gel mixing and injecting?
Okay. So the gel actually forms at body temperature. So methylcellulose is, I don't know who to look at. Methylcellulose is inverse thermal gelling. So that means that it forms a gel as you heat it up. And when you, normally it will gel at a very high temperature of say 90 degrees. And when you mix it with hyaluronan or if you mix it with salts, you can lower that gelation temperature. So it's actually quite easy to mix the cells with the gel and then inject it, and then it just gels at body temperature.
I think everybody is also ready for the break and coffee or tea. So big thank you to all the speakers and chairpersons.